Right, guys, we are now looking at mitosis. Okay, so the purpose of today's lesson really is to draw out these structures and for you to draw them with me. Okay, so the purpose is really to practice drawing mitosis. So, mitosis involves four distinct phases. Okay, to remember those phases, there is a few different kind of phrases that you can remember. One, a very easy one that I use often is pour me another tea. Okay, you can see different examples in your notes to help you. But basically, if you look at the first letter of each of those words, that'll indicate to you what phase you need to draw first. Okay, so the first phase of mitosis is P standing for prophase. Okay, so prophase is the very first step. So I might label that one, and that's prophase. So, what you need to imagine here is what does the cell look like in interphase? Okay, because remember, interphase is just before prophase. So yesterday we talked about a nuclear membrane that was breaking down. We talked about chromosomes starting to form, okay, in the nucleus. And really that's the main thing, really. Okay, so this is continuing in prophase. Now what I'm going to do is I'm going to represent the actual cell membrane here in green, okay, and I'm going to represent the we'll say the nuclear membrane in orange, okay, and I'm going to blow up the nuclear membrane, make it really big so that I have enough room inside in this nucleus, okay, so making it artificially much bigger than what it normally is for the purpose of understanding this. So the nuclear membrane is breaking down, and I want to draw chromosome structures. Now, there's one thing I want to introduce you to, and that is where we have 2n equals 6. So let's say, for argument's sake, this is a Leaving Cert exam question, and I was asked to represent a cell that was diploid and has 6 chromosomes. That would be represented by 2n equals 6. So inside in this nucleus, I need to represent 6 chromosome structures. Now in prophase, these are just randomly located. Okay, so I've now got 5. I need to represent one more. So prophase, what makes it unique? What makes it unique is that the chromosomes are randomly located in the nucleus. The nuclear membrane is breaking down. And that's pretty much the main things that are happening in prophase. Okay. The next thing then is in relation to, and actually, hold on, I forgot to draw 2 structures as well here that are forming. Okay, so what we've got over here on the poles, remember, is we have got the centriole. The centriole has got these fibers that are growing out of it, okay, on either side. Now, if I was to do this correctly, I should have all of these things labeled. So I'm going to label all of this now. And we don't need to worry too much about the cell membrane, but we do need to talk about the nuclear membrane. Okay, and again, we can say nuclear membrane breaking down. That is happening in prophase. Another thing that we want to talk about is the number of chromosomes. So we can say chromosomes. And let's make that smaller again. Chromosomes. We want to talk about the centriole. Okay, so we have the centriole. The centriole is often mixed up for something else called the centrosome. So I hope you remember that the centrosome is located at the center of the chromosomes. We've got the spindle fibers as well. Right, don't forget you should use arrows for this. Now, one common mistake at Leaving Cert level is people don't draw their arrows directly on top of the part that they're describing. So make sure that you accurately draw it in. Okay, and I have a centriole. So I'm going to draw that to this one. Centriole and the spindle fibers. So maybe I'll do the spindle fibers here would be fine. Okay, right. So that is prophase. The next thing then we want to draw is M standing for metaphase. So number 2, metaphase. Now, the next thing that happens after metaphase is that the cell, rather the chromosomes in the nucleus, are starting to line up. Okay, so again, what we need to draw here, I'm just going to draw the cell again. Okay, and I'm going to draw the chromosomes. So I need to draw 6 chromosomes, and they're lined up nicely along the nucleus. So 1, 2, 3, 4, 5, 6. Okay, now the size here, the cell itself doesn't really matter too much, just as long as you have drawn in the structures inside the cell. Okay, you're not being marked on the cell itself, you're being marked on what's happening in the cell. Right, so what we would recognize here if we saw this underneath the microscope is that actually the nuclear membrane is fully gone. Okay, there's no nuclear membrane left. But the centrioles, which are found in opposite locations, they are connected to the chromosomes. Okay, so what these string-like structures, they're connected to the centrosome of each of these chromosomes. So we're going to draw lines here connecting in here to the centrosome of each individual chromosome. Okay, now these, I suppose they're almost like thread-like structures you'd find in a web or something like that. Their purpose, and we'll be talking about that in a bit, is to basically to pull apart these chromosomes. Okay, right, so I've now drawn in the metaphase, but I haven't fully labeled it. Okay, so now just because it takes a little bit longer, I'm not going to label everything, but I might just put a description underneath this for what's really happening. Okay, so what we can say here is that chromosomes are aligned along the equator. Now, this term equator basically describes the center of the cell, center of the nucleus. So that just describes the center. You could say, in addition to that, you could say the spindle fibers are connected at the centrosome. Okay, and that would be, I suppose there's one final point as well I could mention. I could talk about nuclear membrane is gone. Has disappeared. Probably spelled that wrong there. Hold on. Yeah, has disappeared. Right. So those would be the main things here that describe metaphase at that stage. Okay. What we want to move on now is onto stage 3. Okay. So stage 3, which is anaphase. Okay. Now, next stage in anaphase, as I said, the spindle fibers are going to, I just might get rid of this a second or put it to the side. Right. So the spindle fibers are going to actually push or rather pull apart the chromosomes. Okay. So we're going to draw a cell. And this time, remember, we are drawing in our centrioles. This time, however, our chromosomes have been pulled apart. Remember, we have what we call sister chromatids, 2 of them. They're connected together at the centrosome. So what actually has happened here is that these, whoops, I'm going to stick to the red. What has happened here is that these chromosomes are pulled apart. One chromatid goes to the left and one chromatid goes to the right. Okay, so we want to draw in 1, 2, 3, 4, 5, 6. Okay, and the same on this side. 1, 2, 3, 4, 5, 6. And now we want to draw the sister chromatids. Okay, so we have 1, 2, 3, 4, 5, 6 on either side. And what's important here is that these are genetically identical. Sister chromatids are genetically identical. So it's pretty easy to identify this one. What we're saying here is that the spindle fibers have pulled apart. Okay, the chromatids, not to be confused with chromosomes. We could say there is contraction. We could say contraction. So basically, they're pulling towards the centrioles. Okay, that's the main thing there you could talk about there for anaphase. Right, we have one more to go, which is telophase. So T for telophase. Right, so telophase. I always spell that wrong. I think it's telophase. Right, telophase. That is stage 4. Now, at this stage, we have pulled apart the genetics to either pole of the nucleus. What we want to continue now to do is we want to reform the nucleus in this cell, okay? And we want basically two nuclei. So let's move that up a little bit. What we've got next is we have a nuclear membrane reforming on either end of this nucleus, or of this cell rather. Okay, so we have two nuclei. What we should find inside in this is 6 chromatids. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. Okay. What we should also find here is that the spindle fibers have disappeared, and also the centrioles have disappeared. So let's write all of that in. Spindle fibers disappeared, spindle fibers and centrioles disappeared, no longer needed. We also have got, what else did I say? We have nuclear membranes reforming. We've also got 2 nuclei. Okay, so we've got 2 nuclei there for this. Right, let's make that a bit smaller so we can all see that. And it's right next to our diagram. Right, now, of course, this is mitosis now done. It has done its job, which is make 2 identical nuclei. But the next stage here, and this is technically not mitosis, but it is cell division. We call this cytokinesis. Okay, so what's actually going to happen here at the end of this? You might have seen this in the video, by the way, yesterday. What's going to happen next is that the cell needs to basically break apart. Okay, so what happens here? Actually, do you know what? I've made a small little mistake on that previous diagram. I'm just going to make a small little change to that, because what has actually happened there is that something we call a cleavage furrow has started to form. Okay, so actually the cell looks a little bit different to that. It's basically like it's been pinched in the middle, okay, and the cleavage has formed. So what is happening here is cytokinesis, going back to cytokinesis, and basically cells here are splitting apart. Okay, so cytokinesis here, where the cleavage furrow forms and the cells are splitting apart. Okay, so the cleavage will deepen and eventually it'll split fully apart, and what you'll end up with then is 2 separate cells that are genetically identical. Okay, now again, all of these, if you're drawing them on a Leaving Cert paper, should be fully labeled. And once these have split apart, so I'll just do one final diagram, what you'll end up with is 2 cells, 1 and 2. And these 2 cells will have a chromosome number, which is 2n equals 6, 2n equals 6. Okay? Equals. So let's just quickly study the chromosome number. 6 chromosomes here, they're diploid. 6 chromosomes here, they're diploid. We have 12 chromosomes here, okay, in this particular diagram, but that's because there's a copy of each chromosome, so technically it's still 2n equals 6. 2n equals 6. Okay, now over here, the process here of we're going to go through DNA replication to basically copy out these chromosomes to get back to 2n equals 6 to get diploid again. Okay, so look, this has been cell division. I will do a live class on this and basically elaborate on it a little bit more, but that should be an introduction there for you.